Welcome to Bedtime Science. Tonight, consider this. The quiet voice inside you, the one that whispers you're not ready, may be one of the oldest survival tools your ancestors ever developed. Did you know that the feeling of self-doubt activates some of the same brain circuits that once helped early humans avoid predators and social exile? That small, persistent voice, the one that tells you to hold back, to pause, to double-check before you speak, it isn't a flaw. It isn't a sign that something went wrong during your development. It is a quiet guardian, shaped across millions of years, and it lives inside your nervous system right now, doing its work even as you settle in for the night. Tonight we look at why the inner critic exists, not as a mistake in your wiring, but as one of the most carefully preserved features of the human brain. In the next moments, we will gently move through how early social groups rewarded caution over confidence, why the autonomic nervous system, the same system that controls your heartbeat, also controls how safe you feel in a room full of people, how a small brain region called the anterior cingulate cortex acts as an error monitor, scanning your actions before you take them, and why self-doubt, far from being a weakness, may have been the exact trait that kept your lineage alive long enough to produce you. Before we begin, if you find comfort here tonight, consider subscribing to join our quiet community. And if you are still awake, leave a soft comment about what topic we should look at next. Now let us begin. There is a part of your brain that cannot tell the difference between a broken bone and a broken heart. That sounds poetic. It isn't. It's a finding that has been replicated across multiple labs, confirmed with brain imaging, and even tested with over-the-counter medication. In 2003, a research team at UCLA designed an experiment around a simple computer game. They called it Cyberball. The setup was deliberately ordinary. Three players toss a virtual ball back and forth on a screen. The participant in the fMRI scanner believes they're playing with two real people. At first, the ball goes around evenly. Everyone gets a turn. Then, without warning or explanation, the other two players stop, including the participant. They toss the ball only to each other. The participant sits inside the scanner, watching, excluded, left out. What happened inside the brain was worth paying attention to. A region called the dorsal anterior cingulate cortex became noticeably more active during the moments of exclusion. This is a strip of neural tissue folded deep between the two hemispheres of the brain, and it's one of the primary regions that responds to the unpleasantness of physical pain, not the sharp, localized sensation of touching something hot. The deeper, more diffuse quality of pain, the part that makes it feel bad. Social exclusion activated this same region, and the degree of activation matched, with striking precision, how much distress each person reported feeling afterward. The brain was treating a snub in a ball-tossing game the same way it treats a wound. Another region, the right ventral prefrontal cortex, also became active during exclusion, but it played a different role. Its activity went in the opposite direction from distress. It appeared to function as a regulator, a kind of volume dial, working to dampen the alarm signal from the anterior cingulate. The louder the alarm, the harder this prefrontal region worked to turn it down. So even in a simple computer game with no real stakes, no physical contact, no lasting consequence, the brain mounted a full-scale physiological response. An alarm sounded. A regulator tried to quiet it, and the person inside the scanner felt, in a measurable and biological way, hurt. That was just the starting point. 8 years after the cyberball study, a team at the University of Michigan extended the findings in a direction that surprised even the researchers. They recruited 40 people who had recently been through an unwanted romantic breakup, the kind where one person wanted to stay and the other person left. Inside the scanner, participants were shown photographs of their former partners and asked to think about being rejected. In a separate condition, a heated probe was pressed against the forearm, producing controlled physical pain. The researchers then compared the two brain scans side by side. Both conditions activated the dorsal anterior cingulate cortex and the anterior insula. Those are the brain's core circuit for processing the emotional quality of pain, the regions that encode how unpleasant something feels. But the social rejection condition went further than that. It also lit up the secondary somatosensory cortex and the dorsal posterior insula, regions that until then were believed to respond only to physical, bodily pain. The parts of the brain that map where something hurts and how intense the sensation is. The research team compared their activation maps against a database of more than 500 published pain studies. The pattern produced by intense social rejection matched physical pain with up to 88% predictive accuracy. When someone says rejection hurts, they are not using a metaphor. They are describing neural architecture. And this overlap doesn't just show up on brain scans. It responds to medication. A team led by C. Nathan DeWall tested something simple. One group of participants took acetaminophen, the active ingredient in Tylenol, every day for 3 weeks. A second group took a placebo. Over the course of those weeks, the acetaminophen group reported progressively lower levels of daily hurt feelings. The reduction reached statistical significance by day 9. Then came the brain scans. Both groups played cyberball inside an fMRI machine. The acetaminophen group showed reduced activation in the dorsal anterior cingulate cortex and the anterior insula during moments of social exclusion. A physical painkiller, the kind you take for a headache, dulled the sting of being left out of a game. Think about that for a moment. A pill designed for muscle aches also eased social pain, because the brain processes both through overlapping circuitry. The connection runs even deeper, all the way to your DNA. There's a gene called OPRM1 that codes for the mu-opioid receptor, part of your brain's natural pain relief system. One particular variant, the G allele, had already been shown to increase sensitivity to physical pain. Researchers wondered, does this same variant affect sensitivity to social pain? It does. G allele carriers reported higher sensitivity to social rejection. Inside the scanner, they showed greater activation in the dorsal anterior cingulate cortex and the anterior insula during social exclusion. The same gene, the same receptor, governing how strongly you feel both a burn on your skin and a slight from a stranger. So why would the brain be built this way? Why would social rejection and physical injury share the same alarm system? The neuroscientist Naomi Eisenberger offered a clear explanation. Over the course of evolutionary history, the social attachment system, the system that drives you to seek connection, to bond, to remain close to your group, may have piggybacked onto the physical pain system. It borrowed the pain signal, not to cause needless suffering, but to highlight social disconnection and motivate reconnection. Consider what life was like for most of human history. A person alone on an open plain without the shared warmth, shared food, and shared vigilance of a group would not last long. Separation from the group wasn't an inconvenience. It was, for most of our species' existence, a death sentence. So the brain repurposed an alarm that was already working. The signal that once said, "You're injured, pull away from the source," became, also, the signal that said, "You're isolated, move toward the group." And people who lack this sensitivity entirely, who don't feel the discomfort of social disconnection, tend to show patterns associated with personality disorders marked by disordered social bonds and a pull toward isolation. The alarm, uncomfortable as it is, keeps most people tethered to relationships that sustain them. This is the first thread of self-doubt's deep history. The part of your brain that flinches when you sense judgment or exclusion runs on the same wiring that flinches when you touch a flame. It's old, it's biological, and it was selected for, generation after generation, because the ancestors who felt the sting of social rejection corrected course quickly enough to survive. Let that sit. Your brain has a region that monitors for mistakes before you make them. It sits between the two hemispheres, folded into a groove of cortical tissue. It's called the anterior cingulate cortex, and one of its primary jobs is to detect errors, conflicts, and deviations from what was expected, often before you're even consciously aware that something went wrong. The signature of this process has been measured in labs around the world. It's called the error-related negativity, or ERN. It appears as a sharp negative electrical deflection in brain recordings, peaking approximately 50 milliseconds after the commission of an error. 50 milliseconds. That's faster than a blink, faster than conscious thought. The anterior cingulate has already flagged the mistake before the rest of the brain has caught up. In people with anxiety disorders, this signal is amplified. Meta-analyses, studies that pool data from many separate experiments, have consistently shown that anxious individuals produce a larger ERN. Their error monitor runs hotter. It's more reactive. It flags more events as possible mistakes. It whispers "check again" more often and more insistently than the same system in people with lower trait anxiety. Here's the thing, this isn't a malfunction. Not exactly. Picture the environment where this system first took shape. A world of open clearings and hidden predators. A world of rival groups, uncertain food sources, and constant physical danger. In that world, the individual who paused before stepping into an exposed space survived more often than the one who walked forward without hesitation. The one who second-guessed before eating an unfamiliar plant. The one who reconsidered before approaching a stranger. The one who felt a flicker of uncertainty before committing to any irreversible action. That flicker of uncertainty is the anterior cingulate cortex doing exactly what it evolved to do. Patients with damage to this region, lesions that disrupt the anterior cingulate, show diminished ERN signals. Their error monitor runs quieter, and while that might sound like relief from worry, it also means a reduced ability to catch mistakes, to read social cues accurately, to notice when something in the surroundings has shifted in a way that calls for attention. The error monitor is a tool. Self-doubt is what that tool feels like from the inside. Now there's another region involved. Its role is different but deeply linked. The anterior insula sits tucked within the folds of the cortex on the lateral surface of the brain. Its main function is something called interoception, the sense of the internal condition of your body. It reads your heartbeat. It tracks your gut activity, the rhythm of your breathing, the tension in your shoulders, the temperature of your skin. Moment by moment, It builds a conscious map of how your body feels right now. A 2019 study published in the journal eLife showed this directly. When participants focused their attention on sensing their own heartbeat, trying to detect its rhythm without touching their chest, the anterior insular cortex became more active, and the level of that activation predicted how accurate each person was at detecting their own heartbeat. Patients with damage to the anterior insula showed disrupted ability to sense their internal states at all. This region doesn't just passively record the body's signals, it interprets them. And in social situations, it does something very specific. It links what your body is doing to what your mind concludes about safety. When you walk into a room full of people and notice your heart beating faster, your palms growing damp, a tightness forming in your chest, the anterior insula is the part of the brain that brings those sensations into awareness. And once you become aware of them, a second process kicks in. You interpret them. The anterior cingulate, the error monitor, receives the insula's report. It reads the body's arousal in the context of its ongoing scan for threats and mistakes. Something might be off, it concludes. I may have made an error. I may not belong here. Something about this situation doesn't match what I expected. That interpretation feeds back into the autonomic nervous system, increasing arousal. Your heart rate ticks up a little more. Your muscles tighten a fraction further. And the insula reads that increase and reports it again. A loop forms. A 2011 study published in Human Brain Mapping showed this architecture directly. Researchers examined people with spider phobias, measuring brain activity during both a fear-triggering task and a neutral heartbeat detection task. They found clear anatomical overlap in the anterior insula and the dorsal anterior cingulate cortex during both conditions. The same regions processing the phobic fear also process the simple act of sensing one's own heartbeat. Activity in the right anterior insula during heartbeat detection correlated with both trait anxiety scores and state anxiety scores. The better the brain was at reading the body's alarm signals, the more anxious the person tended to feel. This is the architecture of self-doubt, experienced from the inside. The insula reads the body. The anterior cingulate interprets the reading as possible threat. The autonomic nervous system responds with increased arousal. The insula reads that increased arousal. And the cycle continues, each pass reinforcing the one before it. Not a defect, a feedback system. One that, across most of evolutionary history, kept its owners alive by maintaining a baseline of watchfulness in an unpredictable world. Recent laboratory work has begun to identify specific neurochemical keys within this circuit. A study posted on the bioRxiv preprint server found that dopamine D1 receptors in the anterior insula play a direct role in controlling anxiety. When researchers activated D1 neurons in the anterior insula using optogenetics, a technique that uses light pulses to switch specific neurons on and off, anxiety behaviors increased. When they blocked those same receptors, anxiety decreased. The circuit has a chemical switch, and evolution has spent a very long time tuning that switch to a particular setting. Separate work published in Translational Psychiatry offered something hopeful. Interoceptive training, the practice of learning to sense your own heartbeat more accurately, actually altered neural circuits in the anterior insula and reduced both anxiety and physical symptoms of stress. The loop, it seems, can be loosened, not by ignoring what the body is saying, but by learning to read its signals more clearly. When you understand what your body is actually reporting, the error monitor has less reason to sound the alarm. But that skill develops over a lifetime, and the conditions in which it first takes root begin very, very early. In 1940, London was burning. The Blitz, Germany's sustained bombing campaign against British cities, had begun in September of that year. Families across London were being torn apart by evacuation orders, by destroyed homes, by the sheer chaos of wartime life. And in the middle of it, two women set up something unprecedented. Anna Freud, the daughter of Sigmund Freud, and her colleague Dorothy Burlingham established the Hampstead War Nurseries, 3 residential homes funded by the American Foster Parents Plan for War Children, sheltering more than 80 children. The youngest was 10 days old. The oldest was 10 years. What made these nurseries unusual wasn't just the care they provided, it was the observation. Anna Freud was a trained psychoanalyst, and she approached the nurseries as both a caregiver and a scientist. Every child's behavior was recorded in careful detail. Their reactions to air raids, their responses to being separated from parents, their attachment patterns, their emotional shifts across months and years. The result was one of the most detailed records ever assembled of how young children respond to extreme stress, separation, and danger. And the central finding was not what anyone expected. The children were not primarily traumatized by the bombing. Let that land for a moment. Children who stayed with their mothers during air raids, even in extremely dangerous conditions, even with walls shaking and windows shattering around them, showed far less distress than children who had been sent to the safety of the countryside, but separated from their primary caregiver. The critical variable was not how much danger was present, it was whether the mother was there. Anna Freud documented this pattern across 3 books written during and just after the war. She wrote that broken family attachments created more stress in these children than the war itself. The nurseries were eventually reorganized to minimize the disruption of family bonds, providing what she called really good substitutes for the mother relationship. But the deeper observation, the one that matters most for understanding self-doubt, went further than attachment. Children didn't just need their mothers to be present. They absorbed and mirrored their mother's physiological states. When a mother remained calm during an air raid, the child tended to be calm. When a mother panicked, the child panicked, regardless of the actual danger outside. The mother's emotional state functioned as a biological signal. If the mother's nervous system registered safety, the child's system calibrated to safety. If the mother's system registered danger, the child inherited that danger signal, even when nothing threatening was actually happening. This was not conscious imitation. The child wasn't observing the mother, analyzing her behavior, and deciding how to feel. The child's autonomic nervous system was synchronizing with the mother's autonomic nervous system, automatically, below the level of words or thought. The neuroscientist Andrew Huberman, decades later, described this same process in plain terms. He said that the autonomic nervous systems of children tend to mimic the autonomic nervous systems of the primary caregiver. He cited the same tradition of wartime observation that began with Anna Freud in those London nurseries. And modern research has confirmed the mechanism at multiple biological levels, using instruments Anna Freud never had access to. Oxytocin, the bonding hormone, rises in infants after interactions with caregivers. The levels correlate with parent-child affective synchrony. The more attuned the interaction, the higher the oxytocin in both bodies. Salivary alpha amylase, a biomarker of the sympathetic nervous system, the arousal branch, shows a positive correlation between mothers and infants at 6, 12, and 24 months of age. The mother's stress chemistry and the infant's stress chemistry move in parallel. Vagal regulation, the activity of the vagus nerve that governs the calming branch of the nervous system, shows mother-child concordance by age 5. The mother's capacity for calm becomes a kind of template for the child's capacity for calm. Even something as basic as being carried activates the parasympathetic nervous system in both human infants and mouse pups. Distress vocalizations go down, motor activity decreases, heart rate variability increases. The vagus nerve mediates the whole response. The infant's body reads the carrier's body and adjusts its own settings to match. This isn't metaphorical. This is biochemistry. Hormones, nerve activity, and cardiac rhythms all moving in tandem. A study published in 2026 examined 135 Israeli mothers and their children during the first month of an active conflict, when families were exposed to daily missile attacks. The findings echoed Anna Freud's observations from 8 decades earlier. Higher levels of maternal anxiety were positively associated with greater child emotional distress. But this link was not present among mothers with high self-efficacy, mothers who believed they could manage the situation effectively. Those mothers buffered their children from the anxiety transmission. And mothers who talked with their children about what was happening also disrupted the chain. The maternal anxiety-to-child distress connection was only present in families that did not discuss the situation. Here's what that means. When mothers experienced high anxiety but avoided talking about it, children absorbed the stress signal without any framework to make sense of it. The anxiety was transmitted physiologically and emotionally, without explanation, without words. Just one nervous system reading another and calibrating itself to match. The child's body learned to be watchful, not because anything happened to the child directly, but because the caregiver's body was watchful. And that calibration can stick. It becomes a default setting, a resting position on the autonomic seesaw that Huberman describes as the constant interplay between arousal and calm. Parallel research by Renee Spitz in the 1940s documented what happened when this calibration was stripped away entirely. Children separated from their caregivers for extended periods developed what Spitz called anaclitic depression, withdrawal, weight loss, difficulty with sleep, and developmental arrest. If the caregiver returned within 3 to 5 months, recovery was often prompt. After 5 months of separation, the deterioration became increasingly irreversible. Some children did not survive. The attachment bond wasn't just emotional comfort. It was a biological regulator. Remove it, and the child's system lost its reference point. This is the second thread of self-doubt's deep history. The nervous system you carry tonight was not built from nothing. It was calibrated, tuned, by the nervous system of whoever raised you. And their nervous system was calibrated by whoever raised them. And so on, back through generations, each body reading the body before it and adjusting its settings for the world it expected to encounter. Self-doubt, in many cases, is not a conclusion you arrived at through careful reasoning. It's a physiological inheritance, a setting on a dial that was turned before you had words to describe what you felt. That is not damage, it is calibration. The child's brain reads the environment, and for a young child, the environment is primarily the caregiver's body. A child raised in a responsive, consistent household develops a nervous system tuned to expect safety. A child raised in a tense, unpredictable household develops a nervous system tuned to expect threat. Both responses are the brain doing exactly what it was designed to do. The difficulty arises only when the calibration no longer fits the world the person actually lives in. Huberman describes the autonomic nervous system as a seesaw, a constant tilt between sympathetic activation, alertness, readiness, the fight or flight response, and parasympathetic activation, rest, recovery, the feeling of safety. This seesaw tips back and forth throughout the day, and the resting position of that seesaw, where it tends to settle when nothing in particular is happening, is shaped heavily by what was set in early life. He identifies what he calls healthy interdependence. The developmental goal of secure attachment is the capacity for both self-regulation, adjusting your own autonomic state when you're alone, and interactive regulation, being adjusted by the presence of a trusted person. A securely attached adult can calm themselves down in solitude, but they can also be soothed by someone they trust. Both skills are available, and they complement each other. When only one skill develops, the system tilts, and self-doubt often lives in that tilt. The person who can't believe they're safe unless someone confirms it, or the person who can't accept comfort from anyone because their nervous system never learned that another body could be a source of calm. Huberman describes empathy not as a thinking exercise but as a physiological event. He calls it autonomic matching. When two people share personal stories, their heart rates tend to synchronize. The insula, the same region that senses your own internal states, also helps you read the signals of another person's body. It connects interoception, sensing yourself, with the ability to sense someone else. And this brings us to something he calls positive delusion. This isn't a clinical diagnosis. It describes a specific bias that shows up in healthy bonding, the slight overestimation of a partner's qualities that allows attachment to form and hold, the feeling that this particular person can make you feel something no one else can. It's not rational, and it isn't supposed to be. It's a biologically necessary cognitive tilt that stabilizes long-term pair bonds. The brain builds a unique association between one specific partner and internal states of safety, pleasure, and reward, making that bond resistant to alternatives. A deficit in this circuit, a bonding system that runs too cool, may show up as persistent self-doubt in relationships, the inability to believe that someone could genuinely value you, not because the evidence is missing, because the positive delusion circuit isn't generating the gentle bias that makes trust feel possible. Huberman identifies the opposite of positive delusion as contempt. He calls it the antithesis of empathy and the sulfuric acid of relationships, citing research from the Gottman Lab at the University of Washington. Where positive delusion builds bonds by slightly inflating the other person's value, contempt dissolves them by collapsing it. Self-doubt in relationships, then, may not be a character defect. It may be a bonding circuit running below its natural threshold. A system designed to say this person is special, trust them, that instead says be careful, you can't be sure. And like so many features of the cautious mind, this once served a purpose. Bonding too quickly with the wrong person in a setting where betrayal could be fatal was a genuine risk. The cautious bonded slowly. They tested. They waited. Some of them survived because of that waiting. The neurobiologist Alan Score, in conversation with Huberman, described the deeper wiring beneath this system. The attachment relationship, he explained, involves a right brain to right brain connection. The mother, or primary caregiver, intuitively attunes to the infant's emotional states through nonverbal channels, facial expression, voice quality, gesture, timing. This attunement shapes the infant's limbic autonomic circuits in the right hemisphere, forming the foundation for emotional self-regulation that lasts a lifetime. Secure attachment, Score explained, allows efficient switching between calming yourself and seeking comfort from someone else. Insecure attachment creates imbalances. Avoidant individuals over-rely on self-regulation, suppressing the need for others. Anxious individuals chronically seek regulation from others, struggling to find calm within themselves. He noted that a securely attached caregiver is a skilled emotional regulator for the infant, capable of managing both positive and negative emotional states and fostering that same ability in the child. The echo of Anna Freud's London nurseries, 80 years later, translated into the vocabulary of neuroscience. Now let's shift our attention from the individual brain to the social world that shaped it. Picture a troop of chimpanzees, your closest living relatives. Much of a chimpanzee's daily life, particularly for males, involves navigating rank. The social hierarchy is real, and it carries real consequences. Access to food, mating opportunities, safety, grooming partnerships, all of it is mediated by position in the group. And the way rank is communicated is not primarily through violence. It's communicated through ritual. The pant grunt, a rapid, breathy vocalization directed upward in the hierarchy. Field researchers consider it the single most reliable indicator of a subordinate-dominant relationship in chimpanzee society. When a lower-ranking individual encounters a higher-ranking one, this call is produced. It says, "I recognize your position. I am not a threat." The fear grimace, a wide expression with teeth exposed, mouth open, not aggression, appeasement. Bobbing, rhythmic dipping of the head, signaling deference. Presenting, where the animal turns the hindquarters toward a dominant individual, a gesture observed across primate species. And simple avoidance, moving away from higher-ranking individuals before any confrontation can develop. A 2022 study at Chimp Haven Sanctuary recorded behavior during the first 60 minutes of social introduction between unfamiliar chimpanzees. This is a period of intense uncertainty, when no one yet knows where they stand. The most common category of behavior observed was affiliative. Even in a species known for territorial violence, the default response to strangers was social approach. Submissive behaviors came second. Males showed more of them than females during introductions, not because the males were weaker, because they needed to position themselves within the hierarchy quickly, gathering social information as fast as possible. As the researchers put it, males may need to incorporate themselves within the group structure, and that requires learning as much social information as possible in a short time. Submission, in this context, is social intelligence. It's the behavioral expression of the same internal process you experience as self-doubt. The pause that says, "I don't yet know where I stand here, so I will proceed with caution." And the cost of getting it wrong is severe. Observations from GAM, the Tanzanian field site where Jane Goodall spent decades watching chimpanzees, document that a young male who challenges an alpha prematurely may be beaten with genuine violence. In some recorded cases, killed. The males who rise successfully are not the most aggressive. They're the ones who build coalitions over time, groom the right allies, choose their moments with care, and wait. Waiting, assessing, building alliances. These are survival strategies that depend on the capacity for self-doubt, the capacity to say, not yet, not here, not this way. Wolf packs show a parallel pattern. Wolves live in cooperative family units where lower-ranking members contribute to group hunting, territory defense, and pup rearing in exchange for social tolerance. Researchers have described this arrangement as a kind of commodity exchange. And unlike domestic dogs, where low-ranking individuals defer completely over food, subordinate wolves can and do challenge dominant ones over access to resources. The hierarchy is more flexible than it appears from the outside. Some researchers have called it a qualified democracy. Lower-ranking wolves hold what's called leverage power. Their participation in collective survival activities gives them bargaining position. Dominant wolves, in turn, maintain their status not through constant force, but through peaceful sharing. After conflicts, wolves engage in reconciliation behaviors. Bystanders approach victims to offer contact, a behavior labeled consolation. Bystanders also approach aggressors to reduce the likelihood of further attacks. Both patterns maintain group stability. Both require the ability to read social dynamics and respond with restraint rather than impulse. Self-doubt in the social world of our closest animal relatives is not a flaw, it's the operating system. Robert Sapolsky, the Stanford neuroendocrinologist who spent decades studying wild baboons in Kenya, provides some of the most vivid data on what social hierarchy does to the body over time. In stable hierarchies, subordinate male baboons show a clear physiological profile compared to dominant males. High baseline cortisol, the primary stress hormone, elevated resting blood pressure, sluggish endocrine and cardiovascular stress responses, meaning their systems are slower to react to new threats because the baseline is already running hot. Slower recovery after stressors pass, so they remain stressed longer. Reproductive suppression, higher risk of atherosclerosis. Chronic stress, inscribed into the body by social position alone. But here's the detail that matters most for understanding self-doubt. The stress is not driven by objective danger. It's driven by social perception. Sapolsky found that when a male experienced increasing rates of dominance reversals with the male ranked just below him, the threat of being demoted, his baseline cortisol rose. But increasing rates of reversals with the male ranked just above him, the opportunity for promotion, did not raise cortisol. Even though both situations involved equivalent rates of aggression, physical risk, and social disruption. The threat of losing status was stressful. The possibility of gaining it was not. The body responded not to what was happening, but to what it feared might happen next. And low-ranking individuals who simply perceived ambiguous events as threatening, even when other baboons in the same situation didn't react, showed chronically elevated cortisol. There was no actual present-tense threat in those cases. The stress response fired anyway, based on interpretation, based on anticipation, based on a nervous system calibrated to find danger in uncertainty. Sapolsky put it with his characteristic directness. Primates are smart enough and organized just enough to devote their free time to being miserable to each other and stressing each other out. We've evolved to be smart enough to make ourselves sick. A meta-analysis of rank differences in cortisol across primate species found that subordinates showed higher cortisol under two specific conditions. First, when they faced higher rates of harassment. Second, when they had fewer opportunities for social support. Rank alone didn't predict cortisol levels. Rank combined with harassment and isolation did. That maps directly onto human experience. The person who feels self-doubt at work isn't responding to an abstract concept. They're responding to a perceived position in a social order, real or imagined, combined with a sense of how much support surrounds them. The biology is the same. The setting has changed. The savannah is now an office. The baboon troop is now a team. But the cortisol is real, and the anterior cingulate is reading the room in exactly the way it was built to. A 2024 review in Personality Neuroscience traced dominance-related personality traits across primate species and found that these dimensions appear in virtually all primates. They reflect how skilled an individual is at reading and navigating social hierarchies. In a cross-cultural ethnographic study covering 60 human societies, being deferential and respectful toward higher-ranking individuals was recorded as a virtue in 133 separate attestations. It was never recorded as negative, not once, in any of those 60 societies. Deference is universal. Caution in the presence of higher-ranking individuals is universal. The behavioral pattern we call self-doubt shows up everywhere humans organize into groups. One prominent theory of human social evolution holds that our ancestors eventually adopted egalitarianism, but they enforced it through what are called reverse dominance hierarchies, systems where displays of authority were actively punished by the group. Boasting was checked. Arrogance was sanctioned. Even equality required constant social monitoring and restraint. It required the neural machinery that says, watch how you present yourself. Read the room. Don't overreach. Self-doubt is the subjective feeling of that machinery running. Let's turn now to the genetic architecture that keeps this whole system alive across generations. Twin studies, which compare identical twins, sharing all their genes with fraternal twins, sharing roughly half, consistently estimate that 40 to 60% of the variance in neuroticism is heritable. Neuroticism is the personality trait most closely tied to anxiety, worry, and self-doubt. It describes a tendency to feel negative emotions more intensely and more frequently than average. 40 to 60%. That means close to half the variation in how anxious different people feel can be traced to genetic differences that were present at birth. A large meta-analysis spanning all behavioral traits found an average heritability of 49%. Nearly half of the variation in human behavior across every measured dimension carries a genetic component. Genome-wide association studies have identified at least 11 genetic variants linked to neuroticism, some of which overlap with variants connected to depression and anxiety disorders. Common genetic variants currently account for about 15% of neuroticism's total variance, roughly a quarter of the twin study estimate. The rest is likely distributed across thousands of genetic variants, each with a tiny individual effect, collectively shaping how your nervous system responds to uncertainty. The heritability of life satisfaction, how content a person generally feels, sits at approximately 31%, and 65% of that genetic influence flows through personality, mainly neuroticism and extroversion. Your baseline level of worry is in part written into your genome. But that raises a question you might already be asking. If anxiety is costly, if it impairs performance, disrupts relationships, and shortens lifespan through chronic stress, why haven't the genes that promote it been filtered out by natural selection? Shouldn't evolution have cleaned house by now? It hasn't, and the reasons are worth hearing. The evolutionary psychiatrist Randolph Ness proposed what he called the smoke detector principle. Think about the smoke detector in your kitchen. You don't want one that activates only when the house is fully ablaze. You want one that goes off every single time there might be danger. To get that level of reliability, you accept false alarms. Burnt toast triggers it. Steam from the shower triggers it. And you don't replace it with a less sensitive model. Because the one time a real fire breaks out, you need that alarm working. Ness applied the same logic to anxiety. He ran the numbers. If a defensive response, running from a noise in the bushes, costs 100 calories of wasted energy, but failing to run when a predator is actually there costs the equivalent of 10,000 calories in injury or death, then 99% of mathematically optimal defensive responses will be false alarms. 99%. And that's not a malfunction. That's the system working at peak efficiency. Much of the anxiety we feel, Ness wrote, is not a sign of disease. It is the expected output of a system shaped by natural selection to err on the side of caution. He went further. Social anxiety, he observed, is nearly universal in our species because our ancestors who cared what others thought about them consistently did better than those who didn't. The cautious survived. The indifferent often didn't. At a specific gene called SLC18A1, which codes for a protein known as the vesicular monoamine transporter, two variants have been maintained in human populations through a process geneticists call balancing selection. One variant, 136THR, is associated with higher anxiety, depression, and neuroticism. The other, 136IL, is associated with lower levels of these traits. A 2018 study demonstrated that the anxious variant, 136THR, was positively selected during human evolution from ancestral primates. It wasn't a neutral passenger in the genome, drifting along by chance. It was actively favored by natural selection at some point in our lineage. Both variants have coexisted in human populations ever since. This is what balancing selection means. Both versions of the gene persist because each provides advantages under different conditions. In dangerous, unpredictable environments, the anxious variant keeps its carriers watchful and alive. In stable, resource-rich settings, the calmer variant allows its carriers to take risks, explore new territory, and capitalize on opportunities others might pass up. Ness expanded on this directly. An allele that increases anxiety, he noted, will be favored in a dangerous environment. An allele that decreases it will be favored in a safe one. In a world that shifts between danger and safety across generations, which is the world our ancestors actually lived in, both versions persist. Neither eliminates the other. Both are needed. And a 2018 study in the Journal of Psychiatric Research found something that complicates the picture in a fascinating way. Researchers measured anxiety across a continuum and tracked reproductive outcomes, counting children, grandchildren, and great-grandchildren 15 years later. They found that people at both extremes of the anxiety spectrum, both the very cautious and the very bold, left more descendants. People in the middle left fewer. Both ends of the spectrum, not just one. This pattern is called disruptive selection. Evolution is simultaneously preserving both extremes of the anxiety distribution, because both confer reproductive advantages in different roles, different circumstances, different ecological situations. The watchful sentinel who detects threats while others rest, the bold explorer who finds new territory while others hold back. Both are valuable to a group. Both are maintained by the genome. You are not broken for feeling doubt. You carry a particular configuration of a biological system that has been actively maintained by natural selection across the entire history of our species, because it works. But it works for a world that no longer exists. Not quite. The concept of evolutionary mismatch describes the gap between the environment our anxiety systems were built for and the environment we actually inhabit. Sapolsky put it plainly. This is a system that evolved for dealing with short-term physical crises, and we turn it on for chronic psychosocial stress. The ancient brain processes old threats and new ones through different channels. The amygdala and thalamus, the fast, automatic subcortical circuits, respond more quickly to things that would have been dangerous on the savannah. Snakes, heights, angry faces, looming shapes. These get processed before conscious thought even begins. Modern threats, cars, electrical outlets, stock market declines, a passive-aggressive email from your manager, require cortical processing. Slower circuits, more deliberation, less instinct. The anxiety system is still running its original software. It was built for predators and exile. It now fires in response to a text message left on read and a crowded elevator. The alarm is the same. The context has shifted completely. Self-doubt in a meeting is the same neural event as self-doubt on a savanna 200,000 years ago. The anterior cingulate fires its error signal. The insula reads the body's arousal. The feedback loop begins. But in the meeting, there's no predator to evade, no cliff edge to avoid, no hostile stranger who might attack. The alarm sounds into a void, and the person sitting in the chair feels in their body that something is wrong, even though nothing is. This is not a defect in you. It's a mismatch between the tool and the task it's being asked to perform. Now consider what happens when the mind tries to solve self-doubt from the wrong direction. Dr. Alok Kanogia, a psychiatrist who trained at Harvard and works extensively with young adults, draws a clear line between two kinds of self-assurance. He calls them ego and confidence. They sound alike. They function very differently. Ego-driven identity confidence is built on comparison and outcome. It answers the question, am I better than others? It requires constant external validation, and it is, by its very nature, fragile. Kanogia uses a concept from Eastern philosophy called ahamkara, sometimes translated as the I maker. The ego in this framework is everything you reach for to complete the sentence I am, professor, brother, winner, loser, smart, stupid. Each label becomes a piece of identity. Each piece has to be defended. The ego, he says, is not bad. You need it to function, but it can become a cage. Comparison-based desire comes from the ego, and the ego is never satisfied. It always moves the goalposts. Reach the top and now you're watching everyone younger gaining on you. There's no resting state, no arrival point, no quiet. Kanogia describes his own experience. He went from straight A's to straight F's, then became addicted to video games. The collapse wasn't gradual. When the identity, I am a high achiever, met a challenge it couldn't absorb, the entire structure came apart. Because the confidence wasn't rooted in process. It was rooted in outcome. And when the outcome changed, there was nothing left underneath. Self-doubt, looked at this way, actually serves a corrective function. It prevents the catastrophic collapse that comes when an ego-built identity hits genuine failure. The person who carries a gentle current of doubt, who doesn't fully fuse with their successes, has something to fall back on when results don't go their way. The ego is brittle. Doubt, paradoxically, provides a kind of structural flexibility. Process-based confidence is a different thing entirely. It's internal, present tense. It doesn't depend on outcomes or comparison. It answers the question, am I engaged with what I'm doing right now? Not am I the best, just am I here? Canogia offers a simple test. Confidence is a sense of self that lives internally. Ego is a sense of self that lives externally. The ego relies on comparison. Spending time away from other people, he says, builds confidence rather than ego because there's no one to compare yourself with. The feeling of being good enough, he says, is not about what you do on the outside. It's about how you feel on the inside. And you can access this by recognizing the process itself. If nothing feels good enough, that's ego talking. The thought not good enough doesn't need to be obeyed, he says. Just do what you can today. Canogia describes how the ego arises as a protective response to emotional pain. When you experience rejection, when you fail publicly, when someone says no, you feel negative emotions, shame, vulnerability, and the ego steps in as a defense. Its job is to shield you from that pain. It builds a positive shell around the negativity underneath. It inflates. It compensates. It says, I didn't want that anyway, or they just don't get it. The ahamkara constructs a false exterior. And the self-doubt underneath, the raw signal that says that hurt, I might not be enough, gets buried. Not resolved, buried. This connects directly to the evolutionary picture. Self-doubt is the original signal. Ego is the defense that calcifies around it. The problem comes when that defense becomes the identity. When I am successful or I am a failure hardens into a permanent structure rather than remaining what it was meant to be, a passing response to a passing moment. Canogia reframes negative emotions through an evolutionary lens. An emotion, he says, is literally information and motivation. Anxiety carries information. Embarrassment carries information. They aren't just discomfort. They're data from an ancient monitoring system. Embarrassment, he argues, is one of the most useful social tools humans possess. It signals empathy. When you cross a social boundary and feel that flush of embarrassment, you're communicating to yourself and to everyone around you that you recognize the mistake. Being relentlessly confident, the way parts of internet culture encourage people to be, means you stop displaying embarrassment. And that, Canogia says, actually makes you less safe to be around, less trustworthy. Because the people around you can no longer tell whether you understand social norms. Embarrassment is the behavioral expression of the error monitoring system in action. The anterior cingulate flags the misstep. The insula registers the body's flush. And the social signal, the dropped gaze, the sheepish expression, the murmured apology, communicates to the group, I caught it. I know, I'll adjust. Self-doubt expressed in appropriate measure is a signal of social trustworthiness. Canogia also distinguishes between two types of desire that connect to all of this. Sense organ desires are wants that originate from exposure, from seeing something someone else has, from what your eyes or ears encountered on a screen. These are externally programmed. They arrive through comparison. Internal drives are different. They recur across time. They find new objects, new expressions, but the underlying energy stays the same. The same impulse that leads someone to study frogs as a child, then biology, then neuroscience, then start a podcast. The drive doesn't change. Only its expression does. He asks a clarifying question. Is this desire coming from something I saw or from something I've always felt? Self-doubt about sense organ desires may actually be healthy. The doubt is signaling that the desire doesn't originate from within, that it was placed there by exposure, not by intrinsic motivation. The doubt is performing its function. It's saying, check the source of this. And Kanogia makes one more observation that ties together everything we've discussed tonight. Self-esteem, he says, is a being-level thing, not a behavior, not a skill, a substrate, a foundation on which behaviors and skills rest. And when that foundation shifts, everything shifts with it. How you respond to criticism, whether you can hold a boundary, how you interpret ambiguity in someone's tone, all of it changes, not through willpower, through a shift in the ground you stand on. Why not just change the tendency, he asks. That's literally what psychotherapy does every day. Not fighting the inner critic, not drowning it with affirmations or motivational quotes, understanding where it came from, recognizing what it was designed to do, and allowing the deeper substrate, the one that was calibrated in childhood through autonomic co-regulation and attachment patterns, to gradually, gently shift. Not overnight, not through force, through understanding. The behavioral geneticist Catherine Page Harden, at the University of Texas at Austin, has spent her career examining the place where genetics and life outcomes intersect. Her central argument is both straightforward and radical. Genetic differences are not earned. They are not chosen. They are the result of a Mendelian lottery at conception, the random shuffling of chromosomes, the arbitrary assortment of alleles from each parent. You didn't choose your genes for neuroticism any more than you chose your height. These differences contribute meaningfully to variation in psychological traits, academic achievement, and life outcomes. But they are not destiny. As Harden is careful to emphasize, genes and environments interact. Whatever the underlying mechanism may be, behaviors like educational attainment are the outcome of individual tendencies as they are manifested in specific social environments. A genetic tendency toward anxiety can look entirely different in a supportive family than in a chaotic one. The gene is the same. The expression depends on context. And neither genetic advantages nor social advantages are merited. Both are forms of luck. You didn't earn your calm temperament any more than you earned the family you were born into. Self-doubt, then, is partly genetic, partly environmental, partly the product of specific brain regions doing what they were built to do. And the person who carries more of it is not weaker. They're carrying a particular version of a biological system, one maintained by natural selection because, across hundreds of thousands of years, it kept populations alive. Every ancestor in your direct lineage, every single one, stretching back hundreds of thousands of years, survived long enough to have children. That is a mathematical certainty, an unbroken chain reaching back to the earliest members of our species and beyond. Many of them almost certainly carried the same genetic variants for caution and watchfulness that you carry now. The SLC18A1 anxiety allele has been maintained by balancing selection across the span of human evolution. It was positively selected. It didn't persist by accident. Many of them lived in environments where confidence was a luxury and caution was a necessity, where stepping into an exposed space without scanning the edges first was a way to die, where trusting an unfamiliar face without reading intentions first was a way to be exploited, where challenging a stronger individual without securing alliances first was a way to be destroyed. That whisper of doubt, the one that says, "Be careful, check again, you might not be ready," kept them alive. It kept them from walking into clearings where predators waited. It kept them from confronting rivals they couldn't overcome. It kept them from placing trust in the wrong hands at the wrong moment. You are here tonight listening to this because somewhere deep in the past, a person who felt uncertain chose caution and lived. The inner critic is not a flaw in your design. It's a feature, one that was adaptive for most of your species' history. It's running now in a world quite different from the one it was built for. The predators are gone. Exile is unlikely. The open clearing is a conference room. The unfamiliar strangers are colleagues you see 5 days a week. But the circuitry remains. The anterior cingulate still fires its error signal 50 milliseconds after something goes differently than expected. The insula still reads the body's arousal and delivers its report to consciousness. The autonomic nervous system still calibrates to the emotional tone of whoever is nearby. The genes still code for a level of vigilance that was set, in part, by the environments your distant ancestors navigated over thousands upon thousands of generations. Self-doubt is not your enemy. It is a very old voice, doing the best it can to keep you safe in a world it doesn't fully recognize anymore. The environment has shifted. The alarm system hasn't caught up. And the gap between the two, the mismatch between ancient wiring and modern context, is where much of the quiet discomfort of self-doubt originates. But knowing where it comes from changes something. Not everything, but something. When the whisper arrives, you're not ready. They don't really like you. Something is about to go wrong. It can be met differently. Not with frustration, not with the demand that it stop, with a kind of recognition. Oh, that's the error monitor. That's the insula reading my heartbeat and reporting arousal. That's my autonomic nervous system, calibrated long ago, running an old pattern. That's a gene preserved by balancing selection, coding for a trait that kept my ancestors alive. It isn't the truth about this moment. It's a signal from the past, doing the best it can. In stable, predictable environments, confidence pays off. In dangerous and unpredictable ones, caution keeps you alive. Our ancestors lived in the second kind of world for far longer than we have lived in the first. The settings haven't had time to adjust. A few thousand years of civilization cannot override hundreds of thousands of years of calibration. And so the doubt persists, not because something is wrong with you, because the system is working exactly as designed. The question isn't how to eliminate self-doubt, it's how to live with it wisely, how to hear the whisper without automatically obeying it, how to acknowledge the alarm without evacuating the building every time it sounds. That's a different project, a gentler one that begins with understanding. And understanding may have already started tonight. The anterior cingulate cortex right now is easing its monitoring. Not fully. It never fully stops. But as the body settles, as your heart rate slows, as the parasympathetic branch of the autonomic nervous system gradually gains ground over the sympathetic branch, the error monitor reduces its sensitivity. The threshold for alarm rises. Fewer signals pass through. This is what settling in for the night feels like, from the inside. The insula is still reading the body, but the signals it picks up are quieter now. Heartbeat slower, muscles softer, breath deeper. The internal map it's building has shifted from low-grade watchfulness toward gradual release. The autonomic seesaw tips, not abruptly, gently. The sympathetic branch yields to the parasympathetic. Alertness gives way to rest. This is the ancient rhythm, older than language, older than thought, older than the species itself. Somewhere in the genome, the alleles that code for caution and watchfulness are still present. They'll be there tomorrow. They'll be there in the generation after you, and the one after that, maintained by the same selective pressures that have maintained them across hundreds of thousands of years. But right now they are quiet. The neural circuits of social monitoring, the ones that track hierarchy, read facial expressions, calculate risk, and scan for signs of exclusion, are dimming their activity. Not gone, dimming. The way a city's lights lower late at night but never fully extinguish. And the error monitor, that small folded region between the hemispheres that spent the day asking, did I make a mistake? Am I about to make a mistake? Is something wrong? Is lowering its threshold, letting more pass without comment, allowing your mind to drift without interruption. This is the architecture of rest, built from the same materials as the architecture of doubt, the same regions, the same circuits, the same ancient wiring, only the settings have changed. Your brain continues its quiet work. Neurons fire softly in the default mode network, the resting state of the brain, active when the mind is not focused on any external task. Memories consolidate. New connections form between ideas encountered during the day. Even in stillness, the brain is building. The autonomic nervous system adjusts. The heart slows by a beat, then another. The breath lengthens by a fraction of a second. These are not conscious choices. They are the body doing what it has done every night for as long as there have been bodies. Settling, cooling, preparing for the long, slow work of repair that happens only in quiet. Somewhere in the architecture of your mind, the ancient circuits of caution and care still hum, not as a burden, as proof that you come from a long line of survivors, people who doubted, people who paused, people who checked the clearing before they stepped into it, people who read the room before they spoke, people who felt the whisper and listened. You are their continuation. The doubt you carry is their gift, given not in words but in neurons, in genes, in the quiet and persistent calibration of a nervous system that learned over 1,000,000 years to err on the side of keeping you alive. It is an old voice, and tonight it can rest. Sweet dreams, and we will see you next time on Bedtime Science.